Um, Hello, everyone. My name is Emily Herbert, and I'm a PhD student at Drexel University, which is in Philadelphia, in the Material Science and Engineering department. But I am here in Iceland at UNOC with a Fulbright um, research scholarship, and I'm working in a microbiology lab. And so today I'm going to talk about how collaboration between material science and microbial biotechnology can lead to improvements in sustainable bioplastics production. So first, I'm just going to introduce the two fields I'm talking about here. Material science is the study between structure, properties, and processing and performance of solid materials. And typically, the three classes are polymers, metals, and ceramics. And studying material science can lead to certain technologies like batteries and coatings, as well as medical devices and packaging, just to name a few. And microbial biotechnology is the study of living organisms like bacteria, fungi, and yeasts and their derivatives to produce useful products and processes. Some of those products would be like biofuel, small molecules, and polymers. And this has applications in medicine, energy, and wastewater remediation, again, just as some examples. But as you can see, these two fields do overlap with polymer science, and that's what my background is in, so that's what I'm going to talk about moving forward. And polymers are just really, really long molecules that have these repeating structures, and they are pretty ubiquitous in our everyday lives, whether they're from a natural source like cellulose, which can come from different types of plants, or synthetic like polyethylene, which comes from a petrochemical non-renewable source. The truth is that they have some really great properties, and that's what leads to them being used so much and for so many different types of applications. So the polymer that I'm going to talk about that material scientists and microbiologists care a lot about is a class of polymers called polyhydroxyalkanoates, or PHAs for short. And on the bottom of this slide, I have some examples, the most commonly used PHAs, which only have pretty subtle differences in those presence of the length of their side chains. But basically, as an overview, microbes, like, for example, bacteria, will harvest and polymerize carbon from their environment and store these polymers intracellularly as like a carbon reserve. And then they can be grown up at scale and extracted from the polymers, or from the bacteria. So from step 7 on this little graphic, that's where a material scientist would recognize this material and know what to do with it to make whatever they sort of want. So some of the good properties that PHAs have are that they're non-toxic, biodegradable, and very strong. But some of the cons are that they're brittle, difficult to process, and expensive to produce. So sort of the property that I'm going to talk about is the strength of these. The strength and the brittleness go a little bit hand in hand with this class of polymers. And again, it's something that is related both to the microbiology side of things and the material science side of things. So I'm just going to talk briefly about polymer crystallinity and why this is pretty integral to the strength of these polymers and how it can be changed. So, like I mentioned earlier, polymers are really long, and so a material will contain many of these long chains. And you might think that they would stack up in a really ordered way, but the truth is that they behave more like cooked spaghetti, kind of tangling up and folding back on each other. But this does mean that they will arrange in a certain amount of order. So those blue regions where the chains are lining up are called polymer crystals. And those regions are going to contribute to the overall material's strength and hardness, and it has a low permeability of smaller molecules. Whereas the amorphous regions contribute to flexibility, toughness, and permeability of like gases and things like that. So a good example here is a polymer called polyethylene. So the two structures, linear and branched, have the same chemical structure, but a much different kind of architecture. And linear polyethylene has much different properties. It's used for things like rigid plastics like a milk jug, whereas branched polyethylene is much more amorphous, so it's flexible and can be used for things like plastic bags. So the linear polymers are able to pack closely together and form those crystals. And basically this chain alignment really dictates the properties of the final product, and specifically the mechanical properties. Other properties too, but that's just like a good one to think about. So if we think about PHAs and kind of what we can do to not just improve the brittleness, but also provide, a range of properties. Polyethylene is great because you can have different structures and then have different products. Well, we would love if a biodegradable polymer could sort of mirror that. So in order to improve the flexibility of PHAs, a technique to basically break up those crystals, right? So introducing small molecules or those long side chains like branching, like the example with polyethylene, are ways to disrupt the crystallinity, make them a little bit more flexible. So back home in the materials lab, I did a little study where I looked at the introduction of plasticizers, which are those small molecules to break up the periodicity of the polymer chains to try to make them more flexible. And this bottom graph with increasing weight percentages of plasticizer, I get much more flexible films. And I did use some bio-based plasticizers to keep on with that non-toxic trend. But unsurprisingly, we saw also an increase in water vapor permeability of the films, which if you think about something like packaging, that's something to consider, how permeable that film is to small molecules, including water. So this is a well-established relationship, but just kind of highlights some of the limitations of just adding a different molecule. So it's definitely an improvement, but it kind of emphasizes that we also need to be working on the polymer structure side of things, which is where microbial biotechnologists have more tools. So while I'm here at UNAC, I've been working with microbes to try to synthesize PHAs. And that started for me with taking environmental samples in October at Husavik from these tidal pools. And then I introduced them to enrichment cultures, then transferred those to solid plates, where I then isolated the different types of bacteria. So I was only working with one type of bacteria at a time, And then I used a stain called Sudan Black to use as a qualitative indicator of the presence of intracellular polymers within these bacteria. And then once I had some good candidates based on the Sudan Black staining, I did a quantitative measurement of amount of polymer, PHB, in the cells as a function of glucose concentration, which is their main carbon source, what they're converting into polymer. And then I extracted the polymer with something called sock sled extraction. And I wish I had a picture, but I don't. But I have, physical polymer, which is really cool. And that's as far as I've gotten with the characterization of polymer from this batch of bacteria. So I don't know exactly what the structure of the polymer is, but I'm going to find that out soon. And I also have some techniques in the works for how to try to make these branched polymers. So it's definitely in the works, but I still thought that this was just, preliminary work and interesting to highlight the collaboration between these two fields. So with that, I just want to say that the future, I think, of this particular class of polymers in replacing synthetics is continued collaboration between people who have different knowledge and skill set. And I think that the different polymer architectures that are possible are still out there and to be discovered, and as well as different processing techniques that material scientists are more familiar with. So with that, I just want to acknowledge my advisors, my PhD advisor back home, Caroline Schauer and Sean Scully here at UNOC, as well as my lab mates and my funding sources, with a special shout out to Fulbright because that's why I'm in Iceland. So yeah, that's all I got.